Are there lost branches of complex multicellular life within the fossil record? That is the question I want to answer today on Lost Branches of Complex Multicellular Life, Science Spotlight number 6. I'm Dr. Mark Williamson for Transcend. Let's get started. So, as an introduction, we should talk about multicellular life. There are several major extant multicellular eukaryotic lineages. Now, the ones you're probably commonly understand, you recognize from high school biology are animals or metazoans, plants, and fungi. However, there are several other less known lineages across life. This image here is the new Tree of Eukaryotes from a 2020 paper of the same name that goes over the different major groups in Eukaryota. In the past, it was animals, plants, fungi, and then a grab bag called Protists. But really, it turns out Protist was a grab bag. They're divided into supergroups and clades that we can see here today. So, some of the poorly known multicellular eukaryotic lineages are as follows. There's Stamophyla, which is found within the SAR clade. And these are things like multicellular brown algae like kelp. There's also Rhizaria within SAR clade, and there's at least one species of multicellular slime mold within Rhizaria. Over in Archaea plastidia, which includes plants like Chloroplastidia and green algae, there's also the Rhodontophyta, which is the red algae. Over in the excavates, or sort of what was the excavates, there's Discoba. There are different slime molds with limited multicellularity. And then within Amorpha, there are in Amoebozoa, so amoebas and others, there are clades of slime molds with macroscopic multicellular stages of life. And finally, Epistoconta, which includes metazoans, so animals, fungi, and their closest relatives. There is the group called Choanoflagellata, and there are some of those with colonial species, which may or may not, if how you consider it, maybe multicellular, but it certainly complicates trying to figure out what multicellularity means. So with that, it's also good to continue on with a primer and multicellularity. So a good paper on this is Volvox, Chlamydomonas, and the Evolution of Multicellularity in 2010. It has a nice little image here of a tree and different groups of eukaryotes and whether they're multicellular, all of them, some of them, whether they're colonial or not, or whether they're just totally unicellular. And it's really hard to figure out how multicellularity arises. It's hard to investigate, but a useful model system is these two species. So Volvox is a multicellular algae found in images A through, I believe, E, including some of their gamete forms and their normal adult forms. Volvox is a very streamlined, simple type of multicellularity. And then its close relative is an F here, the Chlamydomonas. And so this helps get a feel for like what we might be looking for simple multicellular organisms lost in the realms of time. Another important article that helps line things up is called How Life Got Multicellular, 5 Simple Organisms from 2025, and goes over potential organisms with that. So there are, in the order, there's Salpingoca rosetta, which is a choanoflagellate. They can form a colonial species like we saw earlier. And choanoflagellates are one of the most closest living relatives to metazoans to animals. On the top right here, there's Capsaspora oskarzaki, which is from the Philastrian lineage, which diverged actually earlier than choanoflagellates. Choanoacea flexia is another choanoflagellate. And then there is Spheroforma artica, which is an ichthyosporian, which is a more distant lineage from animals than choanoflagellates and philastrians are. So going even further back in time to figure out how multicellularity works. And then there's Chromocerca Sphaeria perkensi, which is another ichthyosporian. And so one of the big things there is it looks like the things needed for multicellularity occurred several times and were already sort of present in unicellular species. So again, that gives us more of a background, gives us more ammo to figure out where to look for multicellular lineages. And indeed, the evolution of multicellularity is complicated. Turns out that it evolved at least 25 times. That's probably the lower bound. Other articles suggest 40 times. So it certainly happened a lot. Here's one article that talks about that uh, from 2007. And it's found across all major eukaryotic clades. So it shows different clades here and slightly different form what we've seen from sort of stamopiles to excavata to apistocanto, like animals and fungi. And there'll be different proportions of clades that have multicellularity and different types. And so they can be obligate and facultative. So obligate, like us, like animals, we have to be multi-celled, but some can shift between single and multi-cell levels. And this it seems to have risen in the prokaryotes 3 to 3.5 billion years ago in mats of cyanobacteria, because cyanobacteria, bacteria are not multi-celled, they're single cells, prokaryotes, but they can have facultative or sort of colonial organization at times. And truly multicellular eukaryotes occurred around 1.1 or 1.0 billion years ago, and real diversification happened by about 600 to 700 million years ago. And again, multicellularity can take different paths. It can form clonally or by aggregation. So this paper, The Evolution of Multicellularity, a minor major transition, kind of goes over that. That was from 2013. And so there's a difference between again, this paper says type A, aggregative, or type D by division. Division would be clonally. So like animals, why we have multiple cells is, cells in our embryos, divide and grow all together. Whereas other things like slime molds, for example, slime molds can be single cell, but then they sort of clump together in what's called slugs. That's sort of a multicellular, and they can have different split off different cell types or like sporing and things like that. So that would be the aggregative format. And one thing that's interesting, but a little bit beyond the pale here, is that multicellular life had to deal with real issues with like defection and cheating of cells that don't like pull their own weight in either of these setups. All right, so kind of with all that background out of the way, let's get started at looking at some possible lineage, some locations of these lineages in the literature. So we'll start with Ediacaran enigmas. The Ediacaran period happened 575 to 542 million years ago. And this is broken up. The Ediacaran is broken up into three major assemblages. The oldest is the Avalon, followed by the White Sea and Nama. And we find Ediacaran Biota there. This is the first large complex organisms found in the fossil record. And they had all sorts of shapes, from discs to fronds, segmentation, and even these bizarre fractal patterns. For example, brought up in this review, the Ediacaran Biota in 2005. And there's a bunch of different groupings from rangeomorphs all the way to pentaradilomorphs, including things like extant groupings like porifera, so sponges, or things that gave rise to, say, bilateral organisms like bilaterotomorphs. However, with this, I guess there would be two competing theories. One theory is that, all these groups in the Ediacaran biota are sort of precursors to known phyla that we have today. But another more radical view proposed by Dr. Selecher is that all Ediacaran type fossils were because they were more similar to each other, or it looked like they were more similar to each other than any living animals. They're their own group. And so he classified them as an extinct kingdom of life, which he called Vendobionta. So it would be like a non-Metazoan kingdom of complex multicellular life. So that'd be incredibly fascinating if that was true. Now, Is this true? Now, looking kind of further ahead, 2009, there's this review on the eve of animal radiation, which again went over these various groups from rangiomorphs and ernetomorphs being the most interesting. They're having these strange fractal patterns and arising, or at least the rangiomorphs, some of the earliest ones. It kind of looks like that at least some Ediacaran fossils do represent animals that we know of today or precursors. So they have a good picture here where like there's some where actually probably were within the crown group of bilateral animals, so deuterosomes and protostomes. Other ones were probably in the stem group, so somewhere between cnidarians and bilateral animals. Some were definitely within the sponge group, but some might not all of them. Like the rangiomorphs kind of defied traditional phylogenetic interpretation. They might be outside of metazoans altogether. And indeed, this continued to sort of be the case. There's this article, the end of the Ediacaran Biota, again went over these different biota, talked about Vendobionta as a potential hypothesis. And then as late as 2023, in the causes and consequences of end Ediacaran extinctions and update, really went over the fact that while most Ediacaran biota are probably stem group members of extant animal phyla, not all are for sure, especially the rangiomorphs and maybe the ernetomorphs. And so it really seems like some Ediacaran biota might in fact be something like from a Vendobionta, something that's a multicellular non-Metazoan group. So very interesting there. All right, next up, we're going to start looking at sleuthing sterols. So there is Lika. Lika is the last eukaryotic common ancestor that would have arose 1.2 to 1.8 billion years ago. But there's a real big gap between the origin, so Lika, of eukaryotes, and the rise of the crown eukaryotes, the ones that are alive today, the major groups that are alive today, and that actually produce a chemical, a molecule, sterols. So like think of steroids, so they produce sterols. However, we don't see those in between, for large gaps of time. Then in a fairly recent paper, 2023, The Lost World of Complex Life in the Late Rise of the Eukaryotic Crown, we found researchers discovered abundant fossilized what they call protosterol, so be sort of further back on the chain of biosynthesis of sterols. And so they hypothesized that there is this protosterol biota, deep in fossil history. They were present in aquatic systems between 1.6 and 0.8 billion years ago. And so as they say, there seems to be a lost world of ancient stem group eukaryotes that were widespread and possibly abundant during Earth's middle age. And so I guess what that says is there really seems to be very deep lineages of distinct protosterol eukaryotes that we could take a look at. Or not literally take a look at, but hypothesize that might be lost multicellular lineages. So looking a little more closely at this figure, we see on the left here, we see the different ratios of sterols. The purple is the protosterol, so that's what happens. And then the middle here is the branches of different life. We can see on the right, some of the earliest fossil records, including the oldest like red algae, the oldest plants, oldest fungus, oldest amoeba zones, and then right up here would be the Ediacaran biota and Cambrian explosion. And then, so you see in purple is, alongside Luca and before Luca, there's all sorts of these hypothesized protosterol biota lineages that left no living descendants. They aren't around anymore, but might be every bit as diverse as the crown sterile biota we have today. And so a lot of these would have probably been single cell, but maybe not all. Maybe there are clades of protosterol biota that were multicellular. It's a fascinating hypothesis. All right, finally, let's look a little bit later and probably the most fascinating potential lost multicellular lineage in our Prototaxites paradox. So Prototaxites was the first giant terrestrial organism, actually lived on the terrestrial surface, reaching the size of 8 meters in the early Devonian. Now, it's commonly shown as sort of this large mound, and that's what they formed. The taxonomic placement was uncertain. Now, it was fairly quickly ruled out that it wasn't an algae or land plant, so the running hypothesis, the running thought that it was some sort of fungus. However, in this study here, Prototaxites was an extinct lineage of multicellular terrestrial eukaryotes. They really looked at the anatomy and molecular composition, of fossils and found that it really didn't match any known fungus, living or extinct. As they say in the article, no extant group was found to exhibit all the defining features, namely formation of large multicellular structures of varied tube types, a tube composition rich in aromatic phenolic components, and a heterotrophic, likely saprotrophic lifestyle. So their best fit was that actually Prototaxites was from an entirely extinct terrestrial lineage, and that would certainly be a lost multicellular lineage that we're trying to find, trying to talk about. So this is an artist's conception of what Prototaxites might have looked like, and from that article, a really nice table of sort of all these potential groups that they might fall into, different characteristics, and sort of the conclusion of ruling those out, Prototaxites out as one of those groups. So meaning it's something else. All right, so what we were able to look at today was three potential groups that might be lost lineages of multicellular organisms, multicellular eukaryotes. The Vendobionta, especially rangeomorphs or nanomorphs from the Ediacaran Biota, possibly maybe some lineages from the Protosterol Biota, hypothesized, sort of to predate sterol-producing eukaryotes, and then finally, even by the late Devonian, maybe the terrestrial Prototaxites was from a lost lineage of now totally extinct organisms. All right, with that, hopefully that was exciting. Here are all my references that I used throughout the talk. Again, you can leave in the comment what you thought was the most interesting, the most intriguing, and certainly let me know what topic I should highlight next. This is Mark Williamson signing out for Transcend. Thanks.